Now that we've looked at how DNA is structured and replicated, we're going to turn our attention to how DNA actually does what it does. DNA is responsible for carrying all of the genetic information in an organism, but carrying a code is useless if nothing is done with it. Today, we'll look at how DNA is used within an organism at a process called protein synthesis. Now, DNA carries the genetic material for an organism, and that genetic material eventually codes for the synthesis of proteins, but there are a few steps in between. The overall process is called the central dogma, which basically states that DNA codes for RNA, which codes for proteins. We'll start by looking at a bit at RNA. RNA, which stands for ribonucleic acid, is similar to DNA, but with a few major differences. The first difference is in the sugar. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. RNA also uses uracil, which is abbreviated with a U, in place of thymine or T in DNA. When we look at writing mRNA sequences later, we'll see that uracil will bond with adenine, just as thymine did in DNA. And lastly, RNA is usually single-stranded, while DNA is usually double-stranded. There are three major types of RNA that play key roles in protein synthesis. These are messenger RNA, or mRNA, ribosomal RNA, or rRNA, and transfer RNA, or tRNA. Messenger RNA are formed as complementary strands to one strand of DNA, and are responsible for relaying the genetic information from the nucleus to the ribosome to later be turned into proteins. Ribosomal RNA is actually a part of the ribosomes along with proteins. Transfer RNA is made up of smaller segments of RNA, which transport amino acids to the ribosome to form polypeptides. These amino acids are strung together to form the proteins themselves. Here is a comparison of the three major types of RNA. You'll notice the different shapes shown in the models of the three types. The mRNA is important for the code that it stores, so we show the sequence of the individual nucleotides. rRNA's main function is to help make the ribosome function, so we see a bunch of RNAs embedded within the ribosome itself. tRNA's function is to carry and place amino acids, so their shape and structure is the most important aspect. Protein synthesis is divided into two major steps, transcription and translation. Transcription is the process by which mRNA is synthesized from DNA. This occurs in the nucleus and is carried out by an enzyme called RNA polymerase. We'll look at transcription in a bit more detail in another video, but it is worth talking about a bit here. Transcription occurs much like DNA replication, but only one strand of DNA is copied, and instead of being copied into DNA, it is instead transcribed into RNA. This means that thymine is replaced with uracil. When transcription is complete, the mRNA separates from the template strand and the two strands of DNA are bound together again. In this way, transcription can occur without DNA being lost, so one DNA segment can be copied numerous times. Another important part of protein synthesis is the removal of unnecessary non-coding sequences stored in DNA. DNA is made up of mostly non-coding sequences, or sequences of nucleotides that don't actually code for proteins. This might seem like a waste, but it means that if DNA gets damaged, it is a lot less likely to be damaged in a coding sequence, which can help the cell survive even if something manages to damage its DNA. The non-coding sequences are called introns and get removed before the final mRNA is translated into a protein. The coding sequences, called exons, are spliced together to form a strand of mRNA that consists of only coding sequences of nucleotides. Experiments done in the 1960s showed that the DNA code was a 3-base code. What this means is that every 3 nucleotides codes for a specific amino acid. We call these base triplets a codon. Each codon codes for a specific amino acid, though not all codons have unique amino acids. Some codons code for the same amino acid as another codon, as we'll see next. The second major step of protein synthesis is translation, which is the process in which mRNA is translated into an amino acid sequence, forming a protein. Transfer RNA is responsible for putting the proteins together. tRNAs each contain their own 3-base coding sequence, but this one is called an anticodon, as it is complementary to the codon found on the mRNA. Each tRNA carries a specific amino acid, which are lined up and bond together to form a polypeptide. The chart on the right shows the amino acids that each codon codes for. The first base in the 3-base sequence is found on the left, the second base across the middle, and the third base is on the right. For example, a codon with the sequence of UUU would code for phenylalanine. You'll notice that the codon AUG, methionine, is highlighted in blue and labeled START. This is a special codon that tells the RNA polymerase where to start copying mRNA. The 3 highlighted in red are stop codons, which tell the RNA polymerase where to stop. This diagram summarizes the entire process of transcription and translation, starting with the mRNA being transcribed from the template DNA in the nucleus. It then gets transported into the cytoplasm and then the ribosome, where tRNA binds to the codons and begins adding amino acids into a polypeptide sequence, which will later fold into a protein. Experiments done in the 1940s by George Beadle and Edward Tatum led to the one gene, one enzyme hypothesis, which stated that each gene codes for a single enzyme. The experiment was done by growing mutated versions of bread mold on various growth mediums. Although the results of this experiment supported the one gene, one enzyme hypothesis, our understanding of genes has since improved. Some genes encode proteins that are not enzymes, some encode a subunit of a protein that is later combined with other subunits to form a fully functional protein, and still others code for functional RNA that does not get translated into proteins at all, such as rRNA or tRNA. That's all for this video. Make sure to check out the other videos in the classroom as well, and I'll see you next time.